Alright guys, so today we're going to talk about my favorite thing, the immune system. So we're going to focus on the beginning part of the immune system. So this is just kind of understanding the basic structure of how it's set up, how it's broken up into different categories, and how these different categories work together. We'll also talk a lot about the nonspecific host defenses. So these are the ones that are a little simpler, easier to understand. It's basically just the immune system going after anything that it recognizes as completely not self or foreign. The immune system is a little bit in depth. It can be a little confusing. So we will be spending a little more time on this. So Thursday's lecture will be the more in-depth version of the immune system. And I will kind of rehash some of the stuff I'll talk about today to make it a little bit more clear as we go. Okay, so overview of the host immune system. So it's very complex. One of the hardest things to completely understand in the body. It's like trying to understand the universe. I mean, there's just so many complex problems that are associated with the immune system, and the way that it was designed or is designed is just amazing, and you'll see why as we go. Essentially, it's two different systems, but as we study the immune system more and more, we're beginning to realize that they're not necessarily two different systems. They're the same system that have a lot of overlapping features in them, that have one has evolved gradually from the other, but there's still kind of this continuum of bits and pieces in between. So they're not completely segregated, but it makes it a little easier to understand, so it's nice to kind of break it up into these two separate systems. And there are generally three lines of defense, and we'll talk about those as we go. So in general, we're talking about host defenses as one big category. And this is your body, or whichever host you're talking about, whether it be animal or whatever, your body's ability to fight off pathogens or fight off invaders. Anything that comes in, you need a way to get rid of them. And that's what your immune system does. So there's two broad categories, the innate or nonspecific, and the acquired or the specific immune system. The innate or the nonspecific gives you a very quick response, but it is very dumb. It just goes after widely unrecognized things. We'll explain what that means in a little bit. Where the acquired is very smart, but it takes time. But the good thing about it is it has memory. So the innate has no memory. It just recognizes broad categories of things, and it will do that over and over and over again, but it doesn't get any better at it as time goes on. With the acquired, there's specific memory associated with it, and that is what prevents you from getting infected with the exact same thing twice. Within the innate or nonspecific division of the immune system, you have your first line of defense and your second line of defense. Your first line of defense are physical, chemical, and genetic components. So by physical barriers, I mean just physically blocking pathogens from getting in. So things like skin, mucous membranes, and so forth. Chemical barriers aren't alive. That's where the line is drawn. So all of these guys have no living cells associated with them. Instead, chemical barriers, things like lysozymes or enzymes that are secreted that actually kind of break down bacteria, preventing them from setting up shop. And then genetic components are just your DNA, your structure, that makes you either have a weak immune system or a strong immune system. And your immune system is highly variable from individual to individual. And it has been set up that way, primarily because to continue the human race as a whole, you need to have some people that are strong at fighting off organisms or pathogens. And the only way to do that is by constantly changing the immune system from individual to individual. Now, when you're constantly changing the immune system without really knowing how you're changing it, you're going to have a lot of immune systems that aren't going to be that great, because you may change something that actually was a good thing. But you'll have some changes that occur that allow for a very strong immune system. And for that reason, you have people that are immune to things like HIV, or tuberculosis, or leprosy, or plague. And that's what allows the human race to carry on. I know it's a little grandiose and scheme, but it is generally thought of why we have such a varied immune system from individual to individual. Next, we have the second line of defense. So this actually uses cells, and this uses a group of cells called the innate immune cells. And these guys are primarily responsible for phagocytosis, so clearance of organisms. And again, the second line of defense is nonspecific, so it's just looking for broad categories, broad things that are different. We've talked about LPS before. It's only found on gram-negative bacteria. It's never found on humans or human cells. So a lot of these guys have things called TLR4s, which recognize LPS. If they recognize LPS, okay, LPS is never made in a human, so that means it's foreign, gobble it up. But it's not very specific. It's not looking for a specific strain of E. coli. It's just looking for LPS found on all E. coli surfaces. There's also a non-cellular component called complement, which are just special proteins that are circulating throughout your plasma on a fairly regular basis. And they have the ability of attaching and killing non-self pathogens. And I'll explain how this works. It's very complicated. I don't expect you to know every little detail, but I do expect you to know what it is. Then you have things like cytokines that are released. Cytokines are hormones that are released from cells. So they're basically a hormone on a smaller scale. Interferons are one of these. So anytime a cell becomes infected with a virus, they release interferons, and these interferons travel around to their neighbors and warn them that there's a virus present. So it's kind of like a way of communicating. And then the inflammatory response is also part of this innate or non-specific group. And this is where you have these guys phagocytosing things. And when they phagocytose, they get activated, they release cytokines. These cytokines then are released into the periphery and cause inflammation. Now, the third line of defense, or the acquired or specific immune system, is the little bit more complex side of things. And there are two different types of ways of acquiring immunity, either naturally or artificial. So naturally is from either a normal infection or from your mom, from maternal antibodies. So the more common of these is active infection. And this is where you have a whole gamut of adaptive or acquired immune cells, such as B cells, T cells, and the rest, that have the ability of recognizing something very specific as not being self and building a really good immune response to it, an immune response that has memory. But you can also acquire these things artificially. And that's either through a vaccine, so getting vaccinated by maybe inactivated bacteria that cause the infection, and that allows your body to respond and build an immune response to it. Or it can be passive, where you just take someone's antibodies from their body that have already experienced the disease and inject it into someone who has the disease, and those antibodies will help that person fight that infection. So we're going to focus on these two first for today. I don't know why this thing doesn't want to go to the next slide. Okay. So innate, non-specific, you have the first line, second line, acquired and specific, third line. So we'll start off with the innate, obviously. It's the first. It's always on guard, so it's always prepared, and it's quick. But it's not very specific. So that means it doesn't always work the best. And that's why we need the acquired immune response. We need something that can be very specific and can target and hone in on something. It does not improve with repeated exposure, so it has no immunological memory. And also involve inflammatory and phagocytic defenses. And these inflammatory defenses and phagocytic defenses aid in the acquired immune system. So what ends up happening is say if I get infected with an organism, maybe I stab myself with E. coli and it gets into my blood. At first, there's a small amount of organisms. So the immune cells, the innate immune cells, can travel through the blood, through the periphery, go to the organisms, the E. coli, and phagocytose them. Now, as long as the innate immune system can take care of everything, get rid of all the organisms before they start multiplying fairly quickly and becoming too large to handle, that's where the immune system stops. So I cleared out the small amount of organisms, everything's fine. But if for whatever reason it becomes too much for the innate immune system to handle, more innate immune cells will travel to that area. They become activated and they start releasing chemokines and cytokines. And these cytokines result in inflammation and the bringing in of the adaptive immune cells. So the amount of innate immune response determines how serious the infection is going to be and how much your immune system wants to invest into that infection. And if there's a lot of innate immune cells, there's a lot of cytokines being produced, there's going to be a lot of adaptive immune cells that migrate there, and that means you're going to get an adaptive immune response. So first line of defenses, these are the most simple. These are generally physical barriers or chemicals produced by the body to kind of ward off infection. It can be things like pH as well, or salinity conditions, a whole bunch of stuff. Anything that your body kind of changes its physiological environment so that it prevents infection. So you have physical, chemical, and genetic. So the physical barriers are thick skin, things like cilia that kind of push things out or away from the body, mucus, which kind of make bacteria or pathogens get stuck in there, preventing them from doing their job effectively as pathogens, and secretions. So releasing things that kind of break things down, or releasing antibodies that kind of inactivate pathogens. I don't know why this thing won't advance. Okay. Nonspecific chemical defenses, so sebaceous secretions. So this is just releasing oil. By releasing oil, it kind of makes things inhospitable to a lot of pathogens, but there are bacteria that prefer this, and this is what causes acne. Lysozyme and tears. So lysozyme is an enzyme that is produced by tears or in saliva that kind of break down bacteria. Lactic acid. So lactic acid is acidic, making it hard for certain pathogens to survive in. So making an acidic pH or increasing the fatty acid content, make it inhospitable. Increasing hydrochloric acid in the stomach. This kind of breaks down most organisms. Digestive juices and bile in the intestine. So these guys, again, are caustic, inhospitable. Semen, there's antimicrobial chemicals in there, so that all that's in semen is semen, none of the bacteria that are found in the urethra. And acidic pH in the vagina. This prevents things like yeast infections, vaginosis, and so forth. So genetic differences in susceptibility. So some hosts are genetically immune to diseases of other hosts. So everyone's immune system is slightly different, and again, this allows for greater genetic variability in the human population as a whole. So it makes some people have a really good immune system to certain things, and some people have really bad immune systems to other things. And that is a good thing. You want this variability. Alright, so on to the second and third lines of defenses. So the second line is the innate nonspecific, and this deals with actual immune cells. So this is the inflammation, interferon response, phagocytosis, and complement. And we'll talk about these as we go on. The third line, which we'll talk about on Thursday, is the acquired specific, and these produce antibodies as well as activated B cells and activated T cells. So immunology is the study of all the features of the body's second and third line of defenses. So the first line of defenses isn't generally considered immunology, but it does aid in preventing infection. Healthy functioning immune system is responsible for surveillance of the body, so making sure that there are no organisms present that shouldn't be there. Recognition of foreign material, so by doing the way they do this is by recognizing things as not self. So it's impossible for the immune system to say, okay, I know every single thing out in the environment, and I'm going to recognize this as being foreign. It's hard for it to do that. So what it does is it knows everything that the body creates and says, okay, if you don't fit into one of these categories that the body creates, then you're foreign. So it's like process of elimination, not necessarily knowing every single thing that is foreign out there. And destruction of entities deemed to be foreign. So you recognize the foreign pathogen, now you need to clear it, now you need to get rid of it. And it has very effective means of doing this. So the general dogma of the immune system is being able to recognize things as self or non-self. So the way it does this is the white blood cells have the ability of saying, okay, I recognize this specific peptide or protein produced by the immune system or by the body. And if you don't fit into that category, I get rid of you. Oops. And the way that this is done is by a mechanism called tolerance. And that is where immature immune cells are passed throughout the body and are put into contact with special cells that present every single peptide ever produced by your body. And those cells, if they recognize that peptide as being foreign or not self, those cells are killed. So essentially you're weeding out the cells that are going to react to self-made peptides. And this is very important because it's all about the process of elimination. It's your body's ability to say, okay, I've weeded out all of the white blood cells that are going to respond to self, so the only white blood cells that are left are going to be the ones that respond to foreign material. And it evaluates this by examining markers on their surface. So the biggest take-home message from this slide is that you have to be able to differentiate self from non-self. Because if you can't do that, then you're going to just attack everything you see, and that means you'll attack your own body, and it's bad. And this does happen, and when this does happen, you get things like autoimmune disorders. And when you get autoimmune disorders, then you run into a bunch of problems. So systems involved in the immune defenses, so you have body compartments, you have the Reticuloendothelial System, or the RES, which is just a series of connective tissues that allow for these cells to kind of migrate around. The extracellular fluids, so these are the fluids found within or outside of cells, and this again aids in motility of these immune cells, allows them to kind of migrate around. You have the bloodstream, which we've already learned is a pretty effective means of travel. Once you get into one artery or vein, it's pretty easy to travel around the body. And then you also have the lymphatic system, which is the primary compartment for immune cells, and this is the special vessels that kind of mirror where blood, artery, and veins are located, and it allows for immune cells to kind of travel around the body freely. So the RES system is a network of connective tissue fibers, provides a passageway within and between tissues and organs, so it gives something for these immune cells to kind of grab onto and migrate around. The thymus, the lymph node, and spleen are all located within these nice connected pathways, and white blood cells, primarily macrophages, travel up through these. So macrophages are phagocytic cells that are found in tissues. So whenever you're in a tissue, or when you're in the RES system, you're within tissues, obviously, because you're in connective fibers. So components and functions of the lymphatic system. So the lymphatic system is a compartmentalized network of vessels, cells, and specialized accessories and organs. Primarily, it's the lymphatic ducts and the lymphatic nodes. And what these are, the lymphatic ducts, like I've said, mirror arteries and veins, and they collect extracellular fluid. So any extra fluid found in the environment is forced into these tubes, and then these tubes can kind of drain into lymphatic nodes and then drain around the body. Now there is no organ pumping these fluids around. The only thing that allows these fluids to move throughout the body is muscle contraction. So by contracting your muscles, you're kind of putting pressure on those lymphatic ducts, and it kind of forces the fluid through. So it provides a return for extracellular fluid, acts as a drain-off system for the inflammatory response, so it's kind of like a highway of sorts for the immune system to kind of travel around. And it renders surveillance, recognition, and protection against foreign materials. And this is how it aids, or this is because it aids the immune system. So you can see all the lymphatic ducts here, all the lymph nodes, where lymph nodes have a high congregation of immune cells. And during an infection, you can see if I got infected on my foot, it would then travel to these lymph nodes, hang out here. The immune cells would hang out here, they'd become activated, and they could travel further down the lymphatic ducts into the blood and then get around the rest of the body. So lymphatic fluid, also called lymph, it's a plasma-like liquid formed when certain blood components move out of blood vessels into the extracellular space. So it's similar to plasma, but it doesn't have red blood cells. And it has a little bit more albumin in it. It's a little bit more concentrated than plasma. So, lymphatic vessels, again, they follow blood vessels, and that's because they kind of move fluid in between the two. Similar to thin-walled veins, there's a high number in hands, feet, and around the areola of the breasts. You also have a lot within the groin and the digestive tract. Flow of lymph is in one direction only, from extremities towards the heart, and there are valves in place to prevent backflow. And lymph is moved through the contraction of skeletal muscles through which the lymphatic ducts wind their way. So we've talked about lymphatic ducts and lymphatic nodes. Now we'll talk about lymphoid organs. Or we've talked about lymphatic ducts, now we'll talk about lymphoid organs, which lymph nodes are associated with. But as well as the primary lymphatic organs called the thymus and the spleen. So lymph nodes, these are small encapsulated bean-shaped organs. They're basically just like an enlargement of the lymphatic duct. They're found in clusters along the lymphatic channels, large blood vessels, thoracic and abdominal cavities, and they're major aggregations of immune cells. So this is where they kind of hang out, where they talk to each other, where they become activated. The spleen, so it's similar to lymph node, but instead of filtering lymph, it filters blood. And the thymus, so the thymus is a special organ, and this is what allows for maturation of T cells. So T cells are a very specific adaptive immune cell that we'll talk about on Thursday. But what they have the ability of doing is recognizing self and non-self and activating the rest of the immune system. So it's vitally important that you teach these cells what's self and what's not self. And this happens in the thymus. So the thymus is fairly large in a fetus or in a newborn, as you can see in this case. It takes up a good portion. It's about the size of the baby's heart. And as you get older, it atrophies and gets smaller, and that's because your immune system is already established by that point. So when you're a baby, your immune system is very infantile. It's not adapt or grown yet. So it needs to be trained properly, and that's what happens in the thymus. But as you get older, your immune system is fully fledged. It's already been trained. And so the thymus atrophies and gets very, very, very small in adults. And the thymic hormones help thymocytes develop specifically to be released as mature T cells. And there are special cells located in the thymus, and these cells are the ones that produce all the peptides found within the body. And so they're constantly exposing new T cells to these peptides. And as long as the T cell doesn't recognize that peptide as being foreign, the T cell is allowed to leave the thymus and mature and become an active or a naive T cell. As soon as it recognizes these peptides as being non-self, it is then killed within the thymus and not allowed to leave. And then there's also miscellaneous lymphoid tissues, so tonsils are considered enlarged lymph nodes in the throat, breasts of pregnant and lactating women, gut-associated lymphatic tissue or GALT, so your appendix, your lacteals, or pyre patches. Pyre patches are just specialized lymph nodes that are attached to small intestine and large intestine. Mucosal-associated lymphoid tissues are MALT, skin-associated lymphoid tissues are SALT, and bronchial-associated lymphoid tissues or BALT. We like our acronyms in immunology, if you ever notice. So origin, composition, and functions of the blood. So again, we all know blood's found in the circulatory system. It travels throughout. When you look at blood, it's composed of three main components: red blood cells, the buffy coat, and plasma. The buffy coat is basically all the white blood cells. That's where you get this nice little white kind of layer. Plasma is just kind of albumin mixed with water and some antibodies and that kind of thing. That's where the vast majority of the volume of the blood comes from. And then the red blood cells, they're used for carrying oxygen around the body. So serum, so plasma without clotting factors. So plasma has clotting factors in it, obviously. So if you leave it like this, you're going to get all these clotting factors and they can get sticky and kind of clump together if you're doing certain assays. So a lot of times what people will do is just let the blood sit and naturally pool to the bottom. And what will happen is all of these clotting factors will form a nice big clot. And then you'll get serum. So serum is plasma without clotting factors, and it's used a lot in immunology because it carries a lot of antibodies. And a lot of times we're looking at antibodies to see how far your immune system has progressed. And it has a lot of antibodies that don't have clots associated with it. So it makes it easier to use because you're not getting into this sticky, clotting mess. So plasma, its main component is water. Has a lot of proteins like albumins and globulins. Has immunochemicals like cytokines associated with it. Fibrogen and other clotting factors are also found in here. You also get hormones, nutrients, dissolved gases, and waste products. So it's kind of just the fluid that collects everything. So survey blood cells. So blood cells have to go through hematopoesis. This is the production of all blood cells, whether it be red blood cells or white blood cells. They have a relatively short life. So red blood cells live about 28 days. White blood cells, depending on the cell type, can live much longer. So you have red blood cells, white blood cells, and platelets, all produced throughout hematopoiesis, and they go through differentiation. So hematopoiesis are specialized stem cells that are found in the bone marrow, and this, depending on the environment that they're in, dictates what kind of cell type they're going to be, whether they're going to be red blood cells, white blood cells, or platelets. So stages in hematopoiesis. So when you're a very small zygote, or I guess fetus in this case, embryo, you have red blood cells being produced in the yolk sac. Eventually it carries over to the liver. Then as you grow up, become a larger fetus, it starts to be produced in the bone marrow. And then as an adult, all hematopoiesis occurs in the bone marrow. So here's a nice slide that kind of gives you a general synopsis of all the hematopoiesis that occurs. So you start off with a hematopoietic stem cell. Depending on the conditions it's in, it can either go into a lymphoid stem cell or a stem cell for all blood cells. So the lymphoid stem cell can then go into a lymphoblast. These lymphoblasts can then become T cells, B cells, or natural killer cells. The other stem cells, the stem cells for everything but lymphocytes, can then go into 4 different types of stem cells: erythroblasts, megakaryoblast, myeloblasts, or monoblasts. Monoblasts become monocytes, and you'll learn what those are in a little bit. Myeloblasts become neutrophils, basophils, and eosinophils, or granulocytes, or blasts or mast cells, depends on the conditions they're put in. And megakaryocytes kind of break off little portions of their plasma membrane to make platelets, and erythroblasts become red blood cells. So what are the white blood cells composed of? So the vast majority are granulocytes. So these are large cytoplasmic granules, and they're lobed nuclei. That's why they're called granulocytes. You can see all these little dots. The vast majority of white blood cells, about 70%, are neutrophils. And neutrophils, their primary role is just to gobble up junk. So anything they find is foreign or not self, gobbles it up. Basophils and eosinophils aid in things like allergy responses and parasitic pathogen responses. And so their primary role is to kind of wall off organisms and release nasty things that cause allergies, but also piss off parasites. And I'll talk about those in more detail later. Agranulocytes, so these guys have very small granules, but you can't see them with a light microscope, and they usually have a rounded nuclei. So these are the T and the B cells, so these are part of the adaptive immune response, and monocytes. So monocytes are very similar to neutrophils in the fact that they can phagocytose and gobble up things, but they also have the ability to talk to T and B cells after they gobble up things. Neutrophils just gobble things up and don't talk to T or B cells. So neutrophil has a horseshoe polymorphine nuclei, so it's kind of this U shape. Most common leukocyte, so 55 to 90%, so it's the vast majority of cells found in the blood, or white blood cells found in the blood. It's present in high numbers in blood and tissue, and it phagocytoses bacteria, so that's all it does, gobble things up. but it does not have the ability to talk to the adaptive immune response after that. It's just there to clear things, get rid of things. And it's usually the first to arrive, and that's because there's just so many of them. Eosinophils are present in bone marrow and spleen, and these attach and destroy eukaryotic pathogens, so large pathogens like parasites. But if you live in a nice first world country like we do, where we don't come into contact with parasites on a fairly regular basis, they can then be associated with inflammation and allergies. They tend to get bored if they don't come into contact with parasites. So what they tend to do is kind of go after things they shouldn't. Things like pollen or peanuts or something that causes an allergic response. Basophils, so these are the lowest, found in the lowest amount, so about 0.5%. So they function similar to eosinophils. Localized basophils are called mast cells. So when you have a basophil that's found in tissue, it has the ability of becoming primed. So by becoming primed, it means as soon as it comes into contact with its antigen, it then releases all of these nasty things called histamine, and histamine causes allergic responses. So if you have a basophil that's found in tissues that has been primed for something it shouldn't be primed to, maybe like pollen, as soon as you come into contact with pollen, these mast cells release a whole bunch of histamine very quickly, and that's what causes allergic responses or running noses, hay fever, all that kind of stuff. Agranulocytes, so these are the minority of white blood cells, but they're the most important, at least for acquiring an adaptive immune response. So you have your lymphocytes, so this is the second most common leukocyte. Third line of defense, so T cells, cell-mediated immunity, and B cells, antibody-mediated immunity. We'll talk about these more on Thursday. And then monocytes. Monocytes are the guys that work similar to neutrophils in the fact that they can phagocytose, but they have the ability of talking to the adaptive immune cells, or the T's and B cells. So they have the ability of creating an adaptive immune response, or initiating an adaptive immune response. And once they migrate into tissues, they then become macrophages. So monocytes are in the blood or in the periphery. Once they're in tissues, they become macrophages. So monocytes, they're discharged by bone marrow into the bloodstream, and they act as phagocytes for a few days and differentiate into macrophages, or long-lived, once they get into tissues. They're responsible for many specific and non-specific phagocytic and killing functions. So they phagocytose anything that they deem foreign or non-self. Then they process these foreign molecules, present them on their surface, and then lymphocytes can then read these molecules and determine if what they have come into contact with is self or non-self. And they can secrete biologically active compounds that assist, mediate, and attract and inhibit immune cells. So if they're phagocytosing a lot of things that aren't good or are pathogenic or foreign, they start secreting cytokines and chemokines, and this attracts other immune cells, other adaptive immune cells, or more adaptive immune cells, along with creating an immune response like inflammation, fever, all that good stuff. Oops, and then a specialized monocyte or macrophage is called a dendritic cell, and it does this portion, so this processing of foreign molecules and presenting on a surface, much, much more effectively than regular monocytes. So its primary goal is to present to B and T cells, or adaptive immune cells. Let's go, buddy. So, here we have an alveolar macrophage. It's hanging out in and around the lung. So when you go into an alveoli within the lung, there is a resident M cell, or alveolar macrophage, that just hangs out there and grabs anything that comes in. And because our lungs are constantly taking in pathogens, dust, anything that's seen as foreign, these guys are very, very important for the lung immune system. Kind of get rid of this debris. So erythrocytes, so these develop from stem cells in the bone marrow. They lose their nucleus just prior to entering circulation, and they transport oxygen. So these are the red blood cells. Platelets, these are formed elements in circulating blood, not whole cells, they're just little bits and pieces of cells. and they function primarily in homeostasis and releasing chemicals for blood clotting and inflammation. So they're the guys that cause clots. We won't talk about these too much. We learned about these in anatomy and physiology. Alright, so the second line of defense in more detail, like we've talked about before, causes inflammation, phagocytosis, interferon, and then complement. So inflammatory responses, so it's a reaction to any traumatic event in tissues. So as soon as I get pricked in the arm, or I guess in this case in the finger, with a rose thorn, what ends up happening is I kill cells that get pricked, right? I'm breaking through the epidermis and the dermis, and there are plenty of cells that are still alive in those areas, and as soon as that happens, I break through them, I kill them, and they release chemicals, they release cytokines. And these cytokines attract other cells, primarily immune cells, to that area. And once these cells are attracted to this area, they start releasing cytokines, and these cytokines result in swelling, edema, redness, warmth, and pain. So the cytokines that are released are what result in this kind of response, or inflammatory response, or inflammation. And what this does is by producing or increasing the amount of pressure in that area, it's allowing for a greater volume of cells to migrate to that area. And once you get more cells there, the more inflammatory response you can possibly create. And they do this by blocking off lymphatic vessels, or constricting lymphatic vessels, and restricting arterial vessels, so restricting arteries and veins. And by doing this, you increase the amount of liquid in that area, or edema, and you also increase warmth because there's more blood, obviously, and redness because there's more blood as well. So chief functions of inflammation, so mobilize and attract immune components to the site of injury. Set in motion mechanisms to repair tissue damage and localize and clear away harmful substances. So if I increase the amount of blood in a given area, that means I'm increasing the amount of platelets. If I increase the amount of platelets, better chance I have of clotting. And it also destroys microbes and block their further invasion. So if I cut off arteries and veins, I cut off lymphatic vessels, it becomes harder for the microbe to travel through those things to get to the rest of the body. And the most important thing about inflammation, so here I break through the skin, cause injury. This injury kills cells. These cells release cytokines and chemokines. Chemokines are a specialized cytokine that actually attract things to them, and we've talked about these before. As soon as that happens, I attract all these different immune cells, like neutrophils. I've already started to create a clot because my platelets are being congregated in this area. But you can see a lot of neutrophils are kind of infiltrating this area. They start to phagocytose. And as they phagocytose, they're clearing away organisms. But the organism's still growing at that point. So it becomes harder and harder for the neutrophils to clear. So then you start getting things like macrophages and lymphocytes migrating to that area. The macrophages then phagocytose much more of the bacteria. They then process it and present it to the lymphocytes. If the lymphocytes recognize it as not self, they activate all the cells in the area, creating an adaptive immune response. So vascular changes, so how you can control inflammatory responses, and that is by chemical mediators such as cytokines. Once they're released, the endothelial cells in arteries, veins, and lymphodes, or lymphatic ducts, constrict and stop blood flow. And this allows for the red blood cells and the fluid to kind of build up in that area. And lymphocytes have the ability of something called extravasation. So they can actually get through this constricted endothelial cell by kind of splitting through the endothelial cells and breaking into the site of inflammation. And they also release chemokines, so this creates a nice chemical attractant area. So in the area where the actual infection occurred, or the damage occurred, you have the strongest amount of the chemokine, and then as it diffuses through the body, it becomes weaker and weaker and weaker, and the immune cells have the ability of picking up this very faint chemokine scent and following it until it gets stronger, stronger, and stronger, and stronger until they reach the site of infection or damage. So inflammatory responses also cause things like fever. Fever is a systemic response to inflammatory events. So if inflammation gets large enough, if you get enough red blood cells there or white blood cells there, releasing enough chemokines, cytokines, you'll get an inflammatory response. So edema, this is the leakage of vascular fluid into the tissues. This allows for kind of a buildup of fluids, and this buildup of fluids increases the volume in that area, meaning you can have more white blood cells in any given spot, making it easier for the white blood cells to do their job by clearing the infection. It can be clear or serious, or it may contain red blood cells or pus, and pus is where you have a bacteria kind of growing in the fluid, and this creates this kind of murky, kind of debris-filled slush. Diapedesis, so this is how white blood cells leave the blood vessels, also called extravasation, which is the more common term. And this is where the white blood cells can actually get through that constricted endothelial or endothelial cells by kind of extravasating or pushing themselves through their junctions that make up the endothelial cell. And chemotaxis, so this is the white blood cells migrating to a damaged site by following the chemokines. So here you have neutrophils. Over here we have some sort of damage that occurred, which released chemokines. These chemokines then create this nice chemical gradient, and these neutrophils or immune cells can kind of follow this chemical gradient to get to the site of damage or infection. They then extravasate through the endothelial cell. So here you have the endothelial tight junctions between these two endothelial cells, allowing them to get to the site of infection. So benefits of edema. So it dilutes toxic substances. It also allows for fibrin to kind of clot in that area, so it builds up the amount of fibrin present. Allows for phagocytosis. Again, it increases the volume, so it increases the amount of cells that can fit in that area. And it also can attract neutrophils, just by the fact that if you have a lot of fluid there, the bacteria grow in that area because there's more fluid. As soon as they grow, they create more bits and pieces. These bits and pieces allow for neutrophils to migrate to that area. So long-lived inflammation. So short-lived inflammation is good. It allows for clearance of infection. But if you have a chronic infection that goes on for a very long period of time and you have chronic inflammation, it can become a fairly bad thing as well. The more inflammation you have, the more restriction that tissue is going to end up receiving, especially when it comes to blood flow. So that means you're getting less oxygen, which can sometimes result in necrosis, which is bad. Also, if you have chronic infection in a given area and chronic inflammation, it means you're just recruiting more and more and more and more and more red blood cells, trying to get rid of the infection. But if those red blood cells aren't effective at clearing that infectious agent, then you're just going to get larger and larger and larger tissues, larger and larger amount of white blood cells, and eventually these can calcify over and cause things like granulomas, which can be bad. But after, if you have the short-lived inflammation that doesn't last very long, but it has the ability of recruiting macrophages, which then can activate B and T cells, you then get an adaptive immune response, which hopefully will clear a lot of this long-lived inflammatory response. So phagocytosis, so this is the most important bit of the entire immune system. Without phagocytosis, you don't get an adaptive immune response. Without phagocytosis, you don't clear either alive or dead pathogens. So it's very, very important that we have phagocytosis. So the general activities of phagocytes, these are immune cells that primarily phagocytose, is to survey tissue compartments, discover microbes, particulate matter, and injured or dead cells. So their primary role is just to migrate throughout the body on a fairly constant basis, looking for anything that's foreign, not self, or dead. Phagocytose it, process it, and get rid of it. They can also, if you're a macrophage or monocyte, extract immunogenic information or antigens from that material, present it to T or B cells, allowing for an adaptive immune response. So the three main types are neutrophils, monocytes, and macrophages. Neutrophils do not present to adaptive cells, where monocytes and macrophages do. So they're produced in the bone marrow. They then travel through the blood. They extravasate from the blood endothelial tissues and become macrophages in tissue. So phagocytic stages, so they follow chemotaxis. They head towards things called PAMPs. So PAMPs are pathogen-associated molecular patterns. So LPS is a PAMP. It is something that is only found on pathogens. And for that reason, phagocytes can recognize it fairly easily, and they can follow it. And if they find a gradient of LPS, so it's weak in one area and then it gets stronger and stronger and stronger, if they follow that weaker to stronger gradient, eventually they'll get to the area where the LPS is being produced, and that is by bacteria. Once they get there, they bind to these PRR, or pattern recognition receptors, called toll-like receptors. And these toll-like receptors recognize broad categories of pathogens. So like I mentioned before, there's a toll-like receptor called TLR4, which recognizes LPS. So if something's producing LPS, if the toll-like receptor binds to that LPS, it triggers a response in the phagocyte to say, okay, this is foreign because it's producing LPS. My body would never produce that. Let's phagocytose it. It then engulfs, or phagocytose. It then forms a phagosome. So a phagosome is just kind of a internal cavity inside of the phagocyte that has the thing that it phagocytosed in it. It then forms a fusion with the lysosome. This produces a phagolysosome. The lysosome carries acid and enzymes that kind of break down material. So as soon as you form this lysosome-phagosome fusion, you get a release of these acids and enzymes, and it breaks down the material inside of the phagosome. Then you get destruction, or digestion, and then the waste is eliminated. So here's a nice little picture of what goes on. So chemotaxis by the phagocyte, so maybe it's chemotaxing to LPS. Then the LPS binds to these toll-like receptors. So the LPS is found on the bacteria, binds to these toll-like receptors, triggers the phagocyte to bring it in. You get this phagosome forming, full of the bacteria that have just phagocytosed. Lysosomes fuse with the phagosome, making the phagolysosome. The lysosome releases its contents, which are usually enzymes, DNases, RNases, proteases, peroxidases, acids, a whole bunch of stuff. They break down the bacteria. It becomes digested. The guy uses it, the phagosome or phagocyte uses it as a source of nutrients, but then it also releases its debris. So toll-like receptors, these are surface receptors, they bind molecular patterns that are not associated with humans. So they're only found in bacteria or pathogens, things like LPS, for instance. Once these toll-like receptors become activated, it stimulates or activates the phagocyte so that it becomes more phagocytic, and it also releases cytokines, and these cytokines are what initiate the adaptive immune response. So fever, this is an adjunct inflammation. It's an abnormally elevated body temperature. So by elevating the body temperature, you make a more inhospitable environment to a lot of pathogens. So fevers of unknown origin, or FUOs, these can be somewhat hard to figure out, and that's because there's a lot of triggers that cause fever, and if you don't have a specific source of the fever, like maybe a pathogen that's broken through the skin barrier or whatever the case may be, it becomes very hard to pinpoint where it's coming from because almost anything can cause a fever. So initiation of fevers from pyrogens set the hypothalamic thermostat to a higher setting. These pyrogens are released by immune cells. Muscles increase heat production, and peripheral arterioles decrease heat loss through vasoconstriction. So increasing heat makes it more of an inhospitable environment to pathogens. So benefits of fever: inhibits multiplication of temperature-sensitive microorganisms. You guys have learned in lab that most Pathogenic microorganisms prefer to be at 37 degrees. As soon as you increase it to like 38 or 39, it pisses them off, puts them at a disadvantage. It impedes the nutrition of bacteria by reducing the availability of iron. So by increasing the temperature, you're reducing the amount of red blood cells present. By reducing the amount of red blood cells present, you reduce the amount of iron. When you reduce the amount of iron, you reduce bacteria's favorite food source. And it increases metabolism and stimulates immune reactions. So increasing temperature increases the effectiveness or the efficacy of the immune system by increasing its metabolism, increasing its ability to divide and respond. You also have things like antimicrobial proteins that are produced, like interferons. So interferons are a type of cytokine, and they're involved against viruses or intracellular pathogens. So as soon as a cell becomes infected with one of these guys, it triggers the interferon response, which releases interferon alphas, betas, and gammas. And this sends out a signal to the rest of the cells, hey, there's a virus present, get ready. And there are certain cellular mechanisms available to these cells to kind of prevent infection by a virus. And that's what they do. So here you have a cell. It's been infected by a virus. It's releasing interferons. These interferons bind to the nearby cell, and as soon as this cell becomes infected, or maybe it prevents infection by the virus, but as soon as it does become infected, it degrades viral nucleic acids and blocks viral replication, so prevents the virus from infecting the cell. Another antimicrobial protein, or combination of proteins, is something called complement. It's composed of at least 26 blood proteins that work in concert to destroy bacteria and certain viruses. It's basically a cascade reaction that happens automatically. So you have a multitude of proteins that spontaneously hydrolyze, or spontaneously break apart. And when they break apart, they become active. And depending on where they land or what they come into contact with, these active proteins can then bind other active complement proteins and bind more and more and more until they form something called a MAC complex. And this MAC complex can then poke holes in cells. And that's the simplest way that I could explain this, because it's very complex, as you can imagine. There are essentially three different pathways. There's the classical pathway. So the classical pathway requires an adaptive immune response already. And that's because it requires antibody. So you cannot get a classical pathway complement occurring without antibodies present. And you don't get antibodies unless you have an adaptive immune response. So the classical pathway is a way to clear organisms after you already have an adaptive immune response. The lectin and the alternative pathways, on the other hand, do not require an adaptive immune response. They can happen immediately. So the alternative pathway just randomly cleaves, and it binds to cells that are not self. A whole bunch of other stuff binds onto it, forming the MAC complex, which pokes holes in the cell. Now, if you're a human cell, or you're in your host, when these alternative pathway molecules bind to your surface, you have the ability of pulling them internal so that you don't get this MAC complex forming. But if you're a bacteria and don't know any better, it happens and you get pores forming. The cool thing about about complement is that it requires no additional energy. You produce the proteins. Once the proteins are produced, they go on their merry way. They do their thing without any intervention. So the classical, like I mentioned before, requires antibodies. The lectin and alternative does not, but it's not nearly as effective or efficient. This requires something called C3, which is just the name of the protein. It hydrolyzes spontaneously, and this hydrolyzation allows for its active subunit to bind to a susceptible cell, like a pathogen, and then you get a whole bunch of these other C5, C6, C7, C8, C9 binding. Once this happens, it forms a pore in the cell, killing the cell. So here's the complement cascade. This is the classical complement cascade. I'm not going to go through this because I don't expect you to know everything. I just expect you to know that complement are proteins found in the environment or found within the blood. They're produced by the liver, and they result in pores being formed in pathogens. Iron-binding proteins. So iron-binding proteins are important in being able to bind any free iron that is present, because any free iron that is present in the body is very tasty to pathogens. So we have things like hemoglobin transfer and lactoferrin that kind of bind and chelate these iron molecules, preventing them from being readily available to pathogens. And that's it. So hopefully it wasn't too crazy for you guys. Thursday, I'll talk about this stuff in more detail. It will become a little bit more clear if you guys are confused on anything. But hopefully you guys enjoyed the immune system. All right, I will see you Thursday. Bye.